Heme or haem, from Greek alpha mu alpha haima meaning blood, is a cofactor consisting of an Fe2+ ferrous ion contained in the center of a heterocyclic macrocycle organic compound called a porphyrin, made up of 4 pyrrolic groups joined together by methine bridges. Not all porphyrins contain iron, but a substantial fraction of porphyrin-containing metalloproteins have heme as their prosthetic group. These are known as hemoproteins. Hemes are most commonly recognized as components of hemoglobin, the red pigment in blood, but are also found in a number of other biologically important hemoproteins such as myoglobin, cytochrome, catalase, peroxidase, and endothelial nitric oxide synthase. Function. Hemoproteins have diverse biological functions including the transportation of diatomic gases, chemical catalysis, diatomic gas detection, and electron transfer. The heme ion serves as a source sink of electrons during electron transfer or redox chemistry. In peroxidase reactions, the porphyrin molecule also serves as an electron source. In the transportation or detection of diatomic gases, the gas binds to the heme ion. During the detection of diatomic gases, the binding of the gas ligand to the heme ion induces conformational changes in the surrounding protein. In general, diatomic gases only bind to the reduced heme as ferrous Fe2. While most peroxidases cycle between Fe3 and Fe5, and hemoproteins involved in mitochondrial redox, oxidation reduction, cycle between Fe2 and Fe3, it has been speculated that the original evolutionary function of hemoproteins was electron transfer in primitive sulfur-based photosynthesis pathways in ancestral cyanobacteria-like organisms before the appearance of molecular oxygen. Hemoproteins achieve their remarkable functional diversity by modifying the environment of the heme macrocycle within the protein matrix. For example, the ability of hemoglobin to effectively deliver oxygen to tissues is due to specific amino acid residues located near the heme molecule. Hemoglobin reversibly binds to oxygen in the lungs when the pH is high, and the carbon dioxide concentration is low. When the situation is reversed, low pH and high carbon dioxide concentrations, hemoglobin will release oxygen into the tissues. This phenomenon, which states that hemoglobin's oxygen-binding affinity is inversely proportional to both acidity and concentration of carbon dioxide, is known as the Bohr effect. The molecular mechanism behind this effect is the steric organization of the globin chain. A histidine residue, located adjacent to the heme group, becomes positively charged under acidic conditions, which are caused by dissolved CO2 in working muscles, etc., releasing oxygen from the heme group. Types. Major hemes. There are several biologically important kinds of heme. The most common type is heme B. Other important types include heme A and heme C. Isolated hemes are commonly designated by capital letters, while hemes bound to proteins are designated by lowercase letters. Cytochrome refers to the heme A in specific combination with membrane protein forming a portion of cytochrome C oxidase. Other hemes. The following carbon numbering system of porphyrins is an older numbering used by biochemists and not the 1 to 24 numbering system recommended by IUPAC, which is shown in the table above. Heme L is the derivative of heme B, which is covalently attached to the protein of lactoperoxidase, eosinophil peroxidase, and thyroid peroxidase. The addition of peroxide with the GLUTAMYL375 and ASPARTYL225 of lactoperoxidase forms ester bonds between these amino acid residues and the heme 1 and 5 methyl groups, respectively. Similar ester bonds with these two methyl groups are thought to form in eosinophil and thyroid peroxidases. Heme is one important characteristic of animal peroxidases. Plant peroxidases incorporate Heme, lactoperoxidase and eosinophil peroxidase are protective enzymes responsible for the destruction of invading bacteria and virus. Thyroid peroxidase is the enzyme catalyzing the biosynthesis of the important thyroid hormones. Because lactoperoxidase destroys invading organisms in their lungs and excrement, it is thought to be an important protective enzyme. Heme is the derivative of Heme, covalently bound at the active site of myeloperoxidase. Heme contains the two ester bonds at the heme 1 and 5 methyls as in Heme found in other mammalian peroxidases. In addition, a unique sulfonium-ion linkage between the sulfur of a methional amino acid residue and the heme 2 vinyl group is formed, giving this enzyme the unique capability of easily oxidizing chloride and bromide ions. Myeloperoxidase is present in mammalian neutrophils and is responsible for the destruction of invading bacteria and virus. It also synthesizes hypobromite by mistake, which is a known mutagenic compound. Heme D is another derivative of Heme B, but in which the propionic acid side chain at the carbon of position 6, which is also hydroxylated, forms a gamma spirolactone. Ring 3 is also hydroxylated at position 5, in a conformation trans to the new lactone group. Heme D is the site for oxygen reduction to water of many types of bacteria at low oxygen tension. Heme S is related to Heme B by the having a formal group at position 2 in place of the 2 vinyl group. Heme S is found in the hemoglobin of marine worms. The correct structures of Heme B and Heme S were first elucidated by German chemist Hans Fischer.